Something happened in a laboratory that nobody talked about the way they should have. Scientists took saliva samples. They ran them through sequencers. They compared chromosomes. And when the data finally printed out, the researchers had to sit with it for a while before they knew what to say. Because what they were looking at was not just a genetic profile. It was proof that a group of human beings had carried a biological memory for 3,000 years without losing it. That biology had done something that no religion, no government, no army, and no ideology had been able to do across century after century of catastrophe. It had remembered. That is where this story begins. Not in a temple, not in a history book, in a genome sequencer in a laboratory. And what it found will change the way you think about identity, survival, and what it actually means to be human. There are approximately 16.5 million Jewish people alive on this planet right now. That is 0.2% of the global human population. A rounding error, a statistical footnote, a number so small that if you scattered them evenly across every country on Earth, most nations would register barely a trace. And yet, this vanishingly small fraction of humanity has spent the last 2,500 years living inside virtually every major civilization in recorded history. The Roman Empire collapsed. The Byzantine Empire crumbled. The Ottoman Empire dissolved. Every superpower that ever expelled, persecuted, or attempted to erase Jewish communities is now itself a chapter in a history book. The communities kept existing, and all through that impossible timeline, through every forced migration and every massacre and every rebuilding from rubble, something kept traveling with them that nobody could see. Something coded into their cells that would take the invention of modern genomics to finally read. What scientists found when they read it was not what anyone expected. It was stranger than that, more precise, more layered, more human. The first assumption that geneticists brought into this research was the assumption that makes intuitive sense. If a population lives somewhere for 1,000 years, they should start to look like the people they lived with. Ashkenazi Jews had been in Central and Eastern Europe for roughly a millennium. By every normal rule of population genetics, their DNA should have drifted toward German, Polish, Russian profiles. Moroccan Jew, S should look like Berbers. Iranian Jews should look like Persians. Ethiopian Jews should look like other Ethiopians. This is simply how assimilation works. Time plus proximity plus intermarriage plus generation after generation, growing up inside the same geographic and social environment. Populations blend, edges dissolve, origins get diluted until they are almost impossible to separate from the surrounding population. That is the standard story of human migration. Population geneticists at Harvard, Hebrew University, Columbia, and research institutions across Europe sat down expecting to confirm a version of that standard story. What the autosomal DNA data showed them instead was that Ashkenazi Jews, Sephardi Jews, and Mizrahi Jews, communities separated by oceans, by centuries, by completely different spoken languages, food traditions, prayer customs, and cultural frameworks, were still pointing genetically toward the same origin point. Not Europe, not North Africa, not Central Asia, the Levant. That narrow strip of eastern Mediterranean coastline, modern-day Israel, Palestine, Lebanon, Syria, Jordan, the ancient crossroads of 3 continents. 1,000 years of living inside European civilization had left a smaller genetic imprint on Ashkenazi Jews than ancestors they shared with Mizrahi Jews from Iraq and Iran. Ancestors who had lived in a completely different part of the world 2.5 millennia ago. Read that again slowly. 1,000 years left less of a mark than 3,000 years of shared ancestry in a land that most of these people had never personally stood in. That is not what genetics is supposed to do. That is not how time is supposed to work on a genome. And yet, there it was, reproducible, verifiable, peer-reviewed, sitting in the data. What makes this finding hold up under pressure is that it does not come from a single study or a single methodology. Researchers examined the 22 autosomal chromosomes, the non-sex chromosomes that carry the bulk of inherited biological information. They analyzed mitochondrial DNA, the genetic material passed exclusively through maternal lines. They studied Y chromosome markers tracing paternal descent. Every analytical avenue pointed the same direction. Jewish communities globally share more DNA with each other and with other Levantine populations, including Druze and Palestinian Arabs, than they share with the non-Jewish populations they lived among for centuries. The genomic data confirmed what no amount of historical argument had ever been able to settle cleanly. There is a biological thread connecting these geographically scattered communities that runs beneath all the cultural variation, beneath all the regional adaptation, beneath all the languages and the local customs and the centuries of apparent divergence. It runs back to one origin, and science can now measure it. But understanding what shaped that genetic profile requires going deeper into the most extraordinary chapter in this entire story. The Ashkenazi Jewish population, the branch that would eventually account for roughly half of all Jews alive today, carries a genetic history. that has no real parallel in modern population science. It begins with a number, 350. That is the estimated size of the founding population from which all modern Ashkenazi Jews descend. Not thousands, not hundreds of thousands, 350 individuals, a number smaller than a single school building, a number you could fit comfortably in a large lecture hall. Research published in Nature. Communications modeled this bottleneck with precision and found that approximately 600 to 800 years ago, somewhere in medieval Europe, the Ashkenazi Jewish population was compressed to between 330 and 350 people. Half Middle Eastern in their genetic makeup, half European, from those 350 people and the genetic quirks they happened to carry. A population numbering in the millions would grow, and every person in that population would be carrying traces of what those 350 individuals carried. Every single one. The research found that all modern Ashkenazi Jews are related to each other at the level of, at minimum, 30th cousins. Not metaphorically related, not culturally related, genetically related in a way that can be measured with shared DNA segments that researchers can locate and trace across the genome. An entire global population, millions of living human beings, connected by biology to a founding group smaller than most people's extended family. To understand the medieval period that produced this bottleneck, you have to understand what was happening to Jewish communities in Europe during the 12th, 13th, and 14th centuries, the Rhineland massacres, the Crusades moving through communities with lethal efficiency, the Black Death arriving and producing a secondary catastrophe. Because Jewish communities, already isolated by law and social structure, were blamed for the plague itself, violence, forced conversion, expulsion from city after city. The combination of active persecution and enforced social isolation did something to the Ashkenazi gene pool that would echo into the 21st century in ways nobody in that medieval period could have imagined. The random genetic traits carried by whoever survived, whoever remained, whoever rebuilt, those traits did not average out the way they would have in a large, stable population. The EI concentrated, they amplified, they became characteristic. Population geneticists call this the founder effect. And in the Ashkenazi Jewish population, the founder effect produced consequences that are still being measured and diagnosed in modern medical clinics. When a trait carried by some of the 350 founders gets passed through generation after generation of a relatively closed genetic community growing at extraordinary speed, the frequency of that trait in the population climbs far above what you would see in a larger, more genetically diverse group. For certain disease-carrying mutations, this produced outcomes that are specific and measurable and medically significant today. 1 in 27 Ashkenazi Jews carries the Tay-Sachs mutation. In the general global population, the carrier rate is 1 in 250. 1 in 10 carries Gaucher disease mutations, compared to 1 in 100 in non-Jewish populations. Approximately 1 in 40 people of Ashkenazi Jewish origin carries a BRCA gene mutation linked to significantly elevated risk of breast, ovarian, prostate, and pancreatic cancer. In the general population, that rate is 1 in 250. These are not small statistical variations. These are the fingerprints of a founding population left on every generation that followed, amplified by the explosive demographic growth that came after the bottleneck lifted. The Ashkenazi community grew from roughly 50,000 individuals at the beginning of the 15th century to 5 million by the start of the 19th century. 400 years, 50,000 to 5 million. Every one of those 5 million people carrying a genome shaped by the 350 who survived the compression. Every amplification of the founder traits, spreading faster and wider with each generation of that extraordinary growth. But here is where the science moves somewhere unexpected. Researchers noticed that 4 of the most elevated disease mutations in the Ashkenazi population all affect the same biological pathway. Tay-Sachs disease, Gaucher disease, mucolipidosis type 4, and Niemann-Pick disease are all disorders of sphingolipid storage. 4 separate mutations, 4 separate devastating diseases, all clustering around the same metabolic system. That kind of clustering does not happen by random chance. It suggests selection. It suggests that at some point in history, carrying one copy of these mutations, while avoiding the double dose that causes disease, offered some survival advantage, significant enough for natural selection to preserve those mutations across centuries of a closed population. The leading hypothesis points to tuberculosis. Medieval European streets were saturated with tuberculosis for centuries. If heterozygous carriers of sphingolipid storage mutations had even a modest resistance to tuberculosis, natural selection would quietly and invisibly have preserved those mutations in the population, even while the children who inherited 2 copies died from the diseases themselves. Biology does not morality. It optimizes for survival in the current environment. And the medieval environment apparently selected for something that the modern medical environment now diagnoses as a liability. The genetic consequences of surviving medieval Europe are still being discovered in oncology wards and neurology clinics in the 21st century. The NHS in England now operates a specific program offering germline BRCA testing to anyone with at least one Jewish grandparent, formally acknowledging that the elevated mutation frequency in this population is significant enough to warrant population-level screening. The medieval bottleneck is not a historical footnote. It is a living medical reality. The story of the Sephardi Jews adds a different layer entirely. In 1492, the same year Columbus crossed the Atlantic, Ferdinand and Isabella signed the Alhambra Decree expelling all Jews from Spain and Portugal who refused conversion. Communities that had been part of Iberian life for centuries were given months to leave or convert. The ones who left scattered across the Ottoman Empire, North Africa, the Netherlands, the Americas. They carried with them a Judeo-Spanish language called Ladino that is still spoken today in corners of Turkey and Greece and Israel and the Balkans. And they carried their genomes. When geneticists run multidimensional analysis on Sephardi populations, they appear in the data as a cluster positioned genetically between Ashkenazi Jews and North African populations. Not quite either. A genetic intermediate. The Levantine signal is still there, weakened perhaps by centuries of Mediterranean movement, hybridized by contact with the populations they passed through, but unmistakably present. The genome carried something out of Spain that all the enforced conversion and the cultural rupture and the centuries of wandering the Ottoman world could not fully dissolve. Then there are the Mizrahi Jews, and they occupy a different position in this story entirely. These are the Jewish communities of Iraq, Iran, Yemen, Syria, Egypt, and Morocco, communities that in many cases trace a continuous documented presence in those lands back to the Babylonian exile of the 6th century before the Common Era. They did not go to Europe. They did not experience the dramatic medieval bottleneck that compressed and shaped the Ashkenazi genome. They lived for millennia as religious minorities in Arab and Persian cities, Baghdad, Tehran, Cairo, Sana'a, maintaining distinct communal identity, while absorbing some local genetic influence, but holding on to that ancient Levantine core more completely than any other branch. When geneticists examine Mizrahi Jewish genomes, they are looking at what may be the closest living genetic approximation to the ancient Israelite population. The Babylonian exile split the Jewish world approximately 2,500 years ago. One stream went east into the heart of what is now Iraq and Iran. One stream eventually moved west toward Europe. Their modern descendants, the Mizrahi and Ashkenazi populations, show a genetic divergence that has been dated with striking precision to approximately 2,500 years ago. A political catastrophe in the ancient world left a measurable mark on genomes being sequenced in 21st century laboratories. That is what it means when scientists say biology is a historical record. But the most philosophically challenging chapters of this genetic story come from the communities that sit at the edges of every conventional definition. The Beta Israel of Ethiopia are a community that has practiced Judaism in the highlands of East Africa for centuries, possibly millennia. Their form of Judaism developed in complete isolation from the mainstream Jewish world. No Talmud, no rabbinical tradition, a Judaism that appears to have frozen at an earlier stage of development than anything practiced elsewhere, built around the Torah and the Ethiopian Christian calendar and practices that have no parallel in any other Jewish community anywhere on earth. When geneticists analyze Beta Israel DNA, what they find is a genome that is predominantly East African. Their maternal lineages, their paternal lineages, their autosomal profile all cluster far more closely with other Ethiopian populations than with Ashkenazi or Sephardi or Mizrahi Jews. Beta Israel and Indian Jewish communities are considered genetically and culturally distinct from the three mainstream groupings. Their genetic story is primarily the story of the African continent, not the Levant. And yet they practiced Hebrew prayers. They maintained dietary laws. They observed a form of Shabbat. For centuries, in total isolation from any other Jewish community in the world, they kept a religious identity alive that corresponded in recognizable ways to the identity kept alive by communities in Warsaw and Baghdad and Marrakesh and Cochin. That is not a genetic phenomenon. That is something else, something the genome cannot explain and cannot contain. The Bene Israel of Mumbai and the Kochini Jews of Kerala on the southwestern coast of India present a parallel version of the same puzzle. Their own oral tradition traces their origins to Jewish merchants and sailors from ancient Israel, survivors of a shipwreck on the Konkan coast. DNA analysis tells a different story, at least partially. Indian Jewish populations show predominantly indigenous South Asian maternal lineages alongside some shared Jewish genetic markers. Their genome is overwhelmingly South Asian. They look genetically like the populations that have inhabited the Indian subcontinent for thousands of years. And yet they maintained Hebrew liturgy. They preserved Jewish law. They developed distinct local traditions across nearly 2,000 years of complete isolation from any other Jewish community. Traditions so specific and so internally coherent that when contact was finally reestablished with Jewish communities elsewhere, scholars spent decades working out what was continuous and what had evolved independently. Both communities, Ethiopian and Indian, forced the question that the entire field of Jewish population genetics is ultimately circling. What is the relationship between genetic ancestry and cultural or religious identity? The genome gives you one answer, the prayer book gives you another, and they do not always point in the same direction. What makes the Jewish genetic story unusual is not that this tension exists. It is that the tension is so precisely documented, so thoroughly studied, so extensively measured that you can see exactly where the biology and the history pull apart and exactly where they hold together. The study that moved this research to a completely different level came from a team that decided to stop inferring the past from living people's genomes and instead read the past directly. Scientists extracted ancient DNA from physical remains, specifically from teeth pulled from a medieval Jewish cemetery in Erfurt, Germany. Erfurt was home to a substantial Jewish community in the 14th century until a massacre in 1349 during the Black Death, when the community was destroyed and its members killed. The cemetery held the genetic record of that community in their bones. When researchers sequenced the ancient DNA and compared it against modern populations, they found two distinct subgroups buried in the same cemetery. One group carried greater Middle Eastern ancestry. Another carried greater Central and Eastern European ancestry. Two genetically measurable communities sharing the same burial ground, attending the same synagogue, speaking the same language, practicing the same religion, but carrying measurably different genetic profiles. The modern Ashkenazi population, the analysis found, formed from a mixture of these two groups. Roughly 60% of modern Ashkenazi ancestry traces to the subgroup with more Middle Eastern genetic heritage, 40% to the subgroup with more Central European heritage. The population that formed from that merger then absorbed little to no outside genetic influence over the 600 years that followed. 600 years in the middle of Europe, multiple empires rising and falling around them, entire civilizations transforming, and the genetic boundaries of the community held. One finding from the Erfurt study deserves its own moment of attention. Mitochondrial DNA analysis of the remains showed that 1/3 of the individuals in the cemetery descended in their maternal line from a single ancestral woman. One woman, somewhere in the ancestry of that medieval German Jewish community, whose mitochondrial DNA replicated and propagated through the maternal lines until 1/3 of an entire community carried it. That woman lived before the records, before the documentation. She is not in any historical source, but she is in the data. The Erfurt research also confirmed something researchers had suspected. Specific disease causing mutations now widespread in modern Ashkenazi Jews were already present in the medieval population, appearing in the remains at Erfurt, confirming that these mutations came from the original founders, that they were not introduced later, that the bottleneck that produced them was exactly as old and exactly as severe as the genetic models had suggested. Then came something that nobody predicted. A study examining mitochondrial DNA variation across tens of thousands of individuals from East Asian populations and from Jewish communities found that 4 Ashkenazi Jewish individuals carry mitochondrial haplogroups belonging to Eastern Eurasian lineages. The differentiation time of this lineage was estimated at approximately 14,800 years ago. The ancient Silk Road carried more than silk and spices. It carried people. And somewhere in the Ashkenazi genome, a woman from East Asia left a maternal lineage still detectable more than 1,000 years later. The Silk Road is in the genome. A landmark study led by Harry Oster at Albert Einstein College of Medicine analyzed the genes of over 500 Jewish donors from 15 different backgrounds, alongside more than 100 non-Jewish donors. Ashkenazi, Sephardi, and Mizrahi Jews were consistently more genetically similar to each other than to the non-Jewish populations they had lived among. All 3 groups showed Middle Eastern ancestry with varying degrees of regional admixture. The Mizrahi and Ashkenazi divergence dated precisely to approximately 2,500 years ago. A 2020 comprehensive study found that modern Jewish populations derive more than half their ancestry from populations related to the ancient Canaanites, suggesting a continuous biological presence spanning more than 3,000 years. The narrative that Jewish populations descend primarily from converts with no ancient Middle Eastern roots is not supported by the genomic evidence. What the evidence supports is ancient Levantine origin, catastrophic diaspora, partial but incomplete assimilation, and a biological signal that remained legible across timescales that would have erased almost any other population group. The mechanism that kept this genetic signal coherent is not simple and not comfortable. Endogamy, marrying within the community, maintained genetic boundaries across the diaspora. Religious law encouraged it, social pressure reinforced it. But paradoxically, so did persecution. When surrounding societies made intermarriage illegal, when social exclusion made it practically impossible, when ghettoization enforced physical separation, across generations, the persecutors were simultaneously, without intending to and certainly without wanting to, helping preserve the genetic distinctiveness of the population they were trying to eliminate. The forces designed to destroy Jewish communities helped in a specific biological sense to conserve the genomic signal that now allows scientists to identify Jewish ancestry in a saliva sample. The violence meant to erase instead inadvertently preserved. That is one of the stranger ironies that emerges from reading this data carefully. The demographic explosion that followed the end of the worst medieval persecution added another layer of complexity. The Ashkenazi community grew from approximately 50,000 individuals at the beginning of the 15th century to 5 million at the beginning of the 19th. And that growth rate is extraordinary by any demographic standard. And what it meant biologically was that the founder effect traits, the genetic peculiarities that came from those 350 medieval founders, did not stay concentrated in a small population where their consequences might be limited. They spread. They amplified across 5 million people, and then across the millions more who descended from those 5 million. Every founding genetic quirk became more represented, more widespread, more medically significant with each generation of that explosive growth. The genetic consequences of medieval survival became a mass population phenomenon across 4 centuries of demographic expansion. Perhaps the most quietly stunning finding in this entire body of research concerns the people who do not know what their genome contains. Of nearly 2,000 BRCA mutation carriers who provided self-reported ancestry information in one study, 21% did not report Jewish ancestry at all. Yet, of those individuals, more than half carried detectable Ashkenazi Jewish genetic ancestry. Centuries of conversion, of assimilation, of deliberate erasure of family history, of families that changed names and changed religions and changed countries and told their children nothing about where they actually came from. And the biology remained. The DNA carried the information forward, even when the people carrying it had no access to the story. Even when the story had been buried or lost or deliberately hidden, the genome remembered what the history forgot. Families that converted during the Inquisition 500 years ago that built new identities and passed those identities to their children and their grandchildren and their great-grandchildren. Across 5 centuries of accumulated distance from the original rupture, some of those families today are watching a Middle Eastern ancestry signal appear in a commercial DNA test result with no frame of reference to understand what they are looking at. The biology held on longer than the memory. What the science does not show is worth being equally clear about. There is no evidence in any of this research of genetic hierarchy. No evidence of a genetically superior or inferior population. What there is evidence of is something considerably more interesting and considerably more human. A population shaped by an unusual combination of catastrophe and continuity. Bottlenecks so severe they should have erased the population. Growth so explosive it carried every founder trait to mass scale. Persecution so systematic it inadvertently reinforced the very genetic boundaries it was trying to dissolve. Dispersal so complete that communities ended up on every inhabited continent. And despite all of that, a shared biological signal that persisted across the whole of it. The crude frameworks of racial pseudoscience, whether deployed by anti-Semites or by extreme nationalists from any direction, find no support in this data. The actual genetic story is too specific, too layered, too historically contingent, and too thoroughly human to fit inside those frameworks. It refuses them. What this research ultimately reveals is something that extends far beyond any single population. It reveals what biology actually is when you look at it at sufficient resolution. It is a historical record. It is an archive written in base pairs rather than in ink, maintained not by institutions or by scribes, but by reproduction itself, passed through bodies across centuries without anyone choosing to preserve it or knowing it was being preserved. Every human being alive carries that kind of record in their cells. The Jewish genomic story is not unique in having a record. It is unusual in how legible that record is, how distinct, how traceable across such an extended time frame. The specific circumstances of this population's history, the bottlenecks and the persecution and the endogamy and the explosive growth, combine to produce a genetic signal that functions almost like a natural experiment in how identity, ancestry, and biology interact across deep time. Scientists can read it with a resolution that most population histories simply do not allow for. The Erfurt study found that the 2 medieval subgroups eventually merged into a single genetic identity that is now carried by millions of people in America and Israel and Europe and everywhere else the Ashkenazi diaspora eventually reached. Most of those people have never heard of Erfurt. Most of them have no conscious connection to a 14th century German city where their biological history was being written in a cemetery during a plague. But they carry it anyway. In the genome sequencing data published in 2025, researchers confirmed that fewer than 15% of individuals in the Ashkenazi sample carried mitochondrial DNA lineages that entered the population after it was founded. The maternal genetic core is ancient, stable, and predominantly of Middle Eastern origin. The modern analytical tools available to science in 2025 are only confirming with greater precision what earlier studies had indicated. The signal is not weakening. It is being read with increasing clarity. This is where the science reaches the edge of what it can answer. Thousands of people are discovering through commercial DNA tests that they carry Ashkenazi Jewish genetic markers without knowing they had Jewish ancestry. Families that converted under the Spanish Inquisition 5 centuries ago, that built entirely new identities across generations of deliberate distance, are watching Middle Eastern ancestry signals appear in their results, with no family story to explain them. The Beta Israel of Ethiopia maintained a form of Judaism in total isolation, while their genome tells a predominantly African story. The Bene Israel of India prayed in Hebrew for 2,000 years, while their DNA remained overwhelmingly South Asian. The genome and the identity do not always point to the same place, and no amount of additional sequencing will close that gap. What the data does settle, precisely and permanently, is the biological record of survival itself. 3,000 years of catastrophe, compression, and continuity written into human cells in a language that no persecution ever reached, that no expulsion ever erased, that no attempt at assimilation ever fully dissolved. The biology held, and now, for the first time in human history, we can actually read it.